Hello everyone, this is Dr. Horman. Welcome to our first audio recording for genetics. We are going to start discussing Unit 1, which will give us a nice overview of Chapters 2 through 6 and Chapter 8. Now, before I actually start lecturing on the PowerPoints, I just want to make sure that everybody kind of knows where to find all their different tools and resources and how they should approach their notes as we get ready for our first exam. On D2L, you're going to notice that there are folders for each unit. Unit 1 obviously will have all the audio links for your YouTube videos that will have the recorded lectures attached to them. I will be dividing up the lectures so that they're usually not longer than an hour. Part of that is so you have plenty of time to kind of digest the material. And obviously, since it's recorded, you can pause it and take your notes as you go along. You will also find in the folder on D2L a separate PowerPoint. That will be the same PowerPoint that will be featured in the video that you're currently watching / listening to. I highly encourage you to have access to that PowerPoint so that you can go ahead and make your own notations as we move along. In general, what you're going to see for our class is that our exam for Unit 1 and for Unit 2 will be extremely heavily based on the PowerPoints and our recorded lectures. So it is imperative that as you're listening to my lispy voice, if you're not sure about anything or you would like to have a further explanation, to please send me an email or go ahead and note it in the discussion sessions or meet me on Blackboard Collaborative so that I can go ahead and give you more of an elaborative answer so that you're more comfortable with the material. I would also recommend that you definitely read the chapters in the book, and what you can also do as an excellent review is to do the Learn Smart modules indicated as Smart Books on Connect. The Smart Book options has been set up for all the chapters that we're going to be discussing. They will ask you questions as you read the ebook, so it's a really nice way to test your knowledge as you kind of peruse the chapter from beginning to end. Now, without further ado, let's start chit-chatting and let's start taking a look at Chapter 2. Chapter 2 is one of those really nice overview chapters. It's going to take a look at some of the key terms that come into play when you talk about chromosomes. It will also go ahead and give you a section on the cell cycle with an emphasis on the difference between mitosis and meiosis when it comes to cell reproduction. Now, on this particular PowerPoint, I have indicated some of the features that we definitely want to keep an eye on. And I understand that many of you have taken different biology courses beforehand, so some of these might be very familiar to you. Others might look a little distant. Maybe it's something that you want to refresh. And in that case, as we're talking about it, if you still want to get more information, definitely reference back to the lecture book, or obviously you can send me an email and I'll be more than happy to answer your questions or your comments. So we're going to go ahead and take a look at terms such as chromosomes, DNA, histones, nucleosomes, and chromatins. We're going to take a look at some of the differences between prokaryotic and eukaryotic cells. I'm going to show you a karyotype, which you're going to frequently see in different chapters in our course. And obviously, what is the difference between a somatic cell and a gamete? And what does it mean when you say something is diploid versus haploid, autosome versus sex chromosomes? And then I'll also be showing you homologous chromosomes. Homologous chromosomes are another term that you'll frequently run into as we proceed through our chapters, so we definitely want to make sure we have a nice comfort level when we see that terminology. So let's just start with good old-fashioned DNA. I hope that at this point in your academic career, you're comfortable with the fact that DNA, deoxyribonucleic acid, is our genetic material. We know that it is a double helix and it is composed of nucleotides. You have your adenine that tends to bond with your thymine. A goes with T, and you have your guanine that tends to bind with your cytosine. And what the illustration on the left-hand side is trying to showcase to you are all the different ways that DNA can be compacted into the form of chromosomes, what we'll see in our karyotype, which is featured on the right illustration. So let's go ahead and take a look at the top part of our left illustration for our DNA. And what we're seeing here on top, what I'm circling for you, this is good old-fashioned naked DNA. So this is DNA as we're normally seeing it, with the fact that it has the sugar backbone and it has the nucleotides that are basically keeping it together, A with T and G with C. And what we see happening here is that on average, each cell has about 6.5 feet of DNA. It's a massive amount of DNA. So your cells have to figure out a way to compact it as best they can. And that is then where a chromosome comes in. A chromosome is nothing but an extremely long piece of DNA, just compact and condensed to its fullest. In order for us to condense it, we do see that we're going to utilize different steps along the way. So on this illustration, what it shows you is that your naked DNA will eventually go ahead and interact with histones. And histones are going to be storage proteins, and what they'll do is they'll stack up in sections of 8, and the DNA will wrap itself around the histone. And when it does that, then we start calling it a nucleosome. So if you come across the term nucleosome, that is nothing but a strand of naked DNA wrapped around 8 histone proteins. The nucleosomes will continue to stack on top of each other, and remember, the goal is to condense the DNA. And as it stacks on top of each other, what we see happening is it starts to resemble an old school telephone cord, and at that point we call it chromatin. Chromatin is the term that we utilize for various nucleosomes stacked on top of each other. And the chromatin will gather and it will condense and it will continue to coil up until we get our stereotypical X shape that we're used to seeing when we talk about chromosomes. And that's what's featured right over here on our karyotype on the right-hand side. You can see our 46 pairs of, or you can see our 46 chromosomes, or I should say 23 pairs of chromosomes featured right over here on our karyotype. Now, by the time you get to your chromosome level, what we see happening is that oftentimes it will become necessary for the DNA to replicate itself. And when it replicates itself, what we're going to encounter is something called a chromatid. So go ahead and take a look at the bottom of your left illustration, and what you're going to see is you're going to see your chromatin is fully condensed. I'm going to go ahead and draw my little red line through my chromatin. So this is nothing but my coiled up DNA. Here's my little chromatin. And this is what your DNA is going to look like when it's active in the nucleus. So it's doing things like transcription and translation, doing what DNA does. And when it's now time for the DNA to replicate itself, because the cell wants to do cell division, regardless of if it's going to do mitosis or meiosis, we see that the chromatin will make a duplicate copy of itself. I'm going to draw my red line now to the second part, the replicated part. And this replicated part is then described as a chromatid, TID. And a chromatid is just a duplicate part of a chromatin. And oftentimes they'll refer to them as sister chromatids to indicate the fact that they're being held together by a little centromere, and they're being paired up until it's time for them to separate. So notice how at the bottom of the illustration, they've kind of bracketed and they said to you, this is a replicated chromosome because it has 2 chromatins, aka sister chromatids. So please know the difference between coiled up DNA, which is chromatin, and duplicated chromatin, which is going to be your chromatids. Now, your sister chromatids do not always have to be identical copies of each other. In fact, in a little bit, we're going to talk about the fact that when you do mitosis, you basically just photocopy the cell. So in many cases, the sister chromatids are identical. But when you do meiosis, you're able to do crossing over, which is that you're able to swap sections of homologous chromosomes, which means that sometimes the sister chromatins are no longer identical from each other. But that's the story that we're going to follow up in a little bit. Before we move on to the next slide, go ahead and take a look at your karyotype one more time. What you're seeing on your karyotype, as I was mentioning to you before, are the 46 chromosomes that humans have. We can say we have 46, or we could say we have 23 pairs. Of the 23 pairs, 22 of them, they're numbered 1 through 22, based from tallest to shortest. Those are what we like to call our autosomes. Autosomal chromosomes are the same in both males and females. Our 23rd pair is our sex chromosome, and that will either indicate that we have an XX, which is what we see over here, which is then a genetic female, or you have the XY variation, that is then going to be a genetic male. The Y is the little short chromosome over here. Now, our sex chromosome does not get a number. It will just be indicated as either XX or XY. The chromosomes in a karyotype are what they look like when they're in metaphase. So they are all aligned down the middle of the cell. And then what we see here is that you're able to pair them up because, as you know, you get one set of chromosomes from your mom and another set from your dad. So it represents the maternal and the paternal pairing of the 23 chromosomes. Now, another thing I want to highlight that you'll often see, just like when we were looking at the karyotype, is the fact that we can say that we have pairs of chromosomes is because of the contribution of the maternal and the paternal lineage. And what we see happening here is the way we're going to describe the chromosomes that we inherit from both mom and dad is we're going to pair them up and we're going to describe them as a homologous pair. Now, on this illustration right here, they've used the stereotypic colorations. Pink is meant to represent the chromosome that is inherited from mom, and blue is meant to represent the chromosome that you inherit from your father. And as you can see, they have been paired up, so they're the same. So let's, for instance, assume that this is chromosome number 10. You have two of those. One is from your maternal lineage, one is from your paternal lineage. Since both of these chromosomes represent chromosome number 10, we're going to call them a homologous pairs. And a homologous pairs means that they both represent the same chromosome. It does not mean that it's going to have the same type of genes, because what you're going to notice on your pictures is that certain parts of the chromosomes have been highlighted to indicate genes. Now, a gene is a particular section of your DNA that's able to do transcription and translation. So what we see happening is that some of the genes are indicated by a capital letter. Some of them are indicated by a lowercase letter. And that's because it's meant to represent that one version is a dominant with the capital, and the other one is a recessive with a lowercase letter. Both of these genes are what we like to call an allele. It's a variation of the same genomic information. So for instance, this is a very simplistic example. Let's say we were doing a study on hair color, and we discovered that hair color is controlled by the gene B found on chromosome 10. What we would see is that your mother, in this case, would allow you to inherit the dominant version of that gene. So let's say, for instance, the dominant version would be brown hair, whereas your father would give you the version of the recessive allele of that gene. In that case, maybe that would be blonde hair. So both genes will go ahead and give you information about your hair color, but they give you variations. Are you going to have brown hair or blonde hair? And that is then where you have the terminology allele, variations of the gene. As you can see right here, you have also the markings of the sister chromatids, and that is just showing you that it's the chromatin that has been replicated on either side of the chromosomes. So anytime you hear me say a homologous pair of chromosome, that means that we are looking at the pairing of both your maternal and your paternal inheritance. I've also discussed to you the fact that you'll have different genes that will be embedded within the chromosome. Not all of our DNA is able to do transcription and translation, only the sections that are labeled as genes. Now, the location of the gene will often be indicated as a loci. So as you can see on our illustration right here, we have our locational terminology, and it's marking gene A, B, and C. And it also highlights the fact that even though you have a homologous pair, so the maternal and the paternal contribution, not all genes are created equal. So you can see, for instance, that in this case, this individual inherited both dominant versions of A from both parents, but when it comes to gene B, they inherit a recessive version from mom and a dominant version from dad. And if it comes to gene C right here, both parents went ahead and contribute their recessive version. Now, a dominant version of the gene usually indicates that that particular genotype will be present in the phenotype or the appearance or behavioral trait of the individual, whereas the recessive trait tends to be masked or hidden, and it will only be apparent in the phenotype if both copies that are inherited are recessive. And that is because the dominant is able to mask the recessive gene. Since we are a diploid organism, meaning that we inherit two copies of everything, one from mom and one from dad, we often have to describe if those copies that we inherited are going to be the same or different. So for instance, right over here at the bottom of my illustration, it says that when you take a look at gene A, B, and C, you can describe the genotype, the DNA that you inherit. Your option will either be that they're both the same or that they're different. If they're the same, we like to say that's homozygous. A homozygous pairing means that both of them are the same. In this case, for letter A, they're both homozygous dominant. If we come over here to C, you'll notice that both of the C's are lowercase, and that's because they are recessive. And because they're the same, once again, you're going to notice that they're homozygous recessive. If you inherit one of each, then you're looking at a heterozygous mix. It is very important to familiarize yourself with heterozygous and homozygous descriptions when it comes to the genotype, because that will help us as we start perusing through our Punnett Squares. So if you need to go ahead and do a little bit more additional readings when it comes to homozygous and heterozygous, please refer to Chapter 2 of the book. The other thing that we see mentioned in Chapter 2 is good old-fashioned cell division, right? How do you maintain the number of cells that you have in your body? How do you go ahead and repair some of the damaged or old ones? And what we see happening here is that we can often utilize either mitosis or meiosis. When you discuss cell division, you often also see a wide difference between a eukaryotic and a prokaryotic cell. Prokaryotic cells are going to be cells that lack a nucleus. This is usually what you see in very unicellular simplistic organisms such as bacteria. And when those cells tend to replicate, what they'll tend to do is they'll do binary fission, which is a form of asexual reproduction, which really at the core is just mitosis. What we see happening in prokaryotic cells is that the DNA will go ahead and replicate itself. And whenever the DNA is replicated within the cell will go ahead and divide itself. It will do cytokinesis, divide its cytoplasmic content, and the end result is that you go from having one cell to two daughter cells. And the two daughter cells are genetically identical to the parent cell. And that is because there is only one source of DNA that was introduced in the mother cell. When it comes to eukaryotic cells, it turns out that we will have a little bit of variety. We can either have mitosis or meiosis, and part of that depends on if you want to make sure that the genomic material in the daughter cells is identical, or if you want to reduce the amount of chromosomes that they carry. In order to discuss the cell cycle, we're going to quickly take a look at what it means when you go through the different phases, including interphase, prophase, metaphase, anaphase, and telophase. Now, your prophase, metaphase, anaphase, and telophase, those will be part of the cell division part, and we definitely have to take a look if we're going to be doing mitosis or meiosis. All right, so here's just a really quick illustration showing you your cell cycle. And as you can see right here, it's often indicated as a circle because it is a continuous state. And what we see happening is that the majority of the cell's life is spent in this darker blue section in what we call interphase. Interphase is day-to-day cell activity. It's divided into G1, S, and G2. G1 is usually when you have a brand new cell, and the cell will be interested in growing, and it will start initiating transcription and doing its day-to-day function. When the cell migrates over to the S phase, its most important job is to synthesize. That's what the S stands for. It stands for synthesis. And in this case, what we're synthesizing is an extra copy of our DNA. So for instance, if we're looking at a skin cell, when the skin cell enters S phase, that is then when we see that the 46 chromosomes that we have in our skin cells will duplicate each other, will replicate each other, and will go from 46 to 92. And the reason that you are increasing your DNA load is because eventually you're going to get into the cell cycle or cell division section of the cell cycle, which will allow us to increase the number of cells that we have. After the DNA has been replicated in S phase, the cell will go ahead and move into G2, and that just stands for gap phase or growth phase 2. And all we're doing here is making sure that there's no mutations and that the cell is ready to proceed into the cell division section. The cell division section is when you are actively going to create your daughter cells. Now we're going to start off taking a look at mitosis. The goal of mitosis is to go ahead and produce two daughter cells that are identical when it comes to its DNA and chromosome to the mother cell that was generated. So you're going to go from one mother cell, to two daughter cells that are going to be identical in their DNA and chromosome composition. In order to do mitosis successfully, you do have different phases that come into play, and that's what you see here separated on the cell cycle. So we're going to quickly take a look at these phases, including what is happening in prophase, prometaphase, metaphase, anaphase, and telophase. And a really quick mnemonic to remember: interphase, prophase, metaphase, anaphase, and telophase in order is just remember the sentence, I passed my anatomy test. And keep in mind that when you're saying that I passed my anatomy test, you're telling me all the different stages of the cell cycle, so the entire thing. If you were only to tell me the stages of mitosis or your cell division, you would not include interphase, because interphase is just day-to-day activity. If you were to tell me what's happening in mitosis, you would start with prophase and work your way all the way down to telophase. Alrighty, so here we are looking at an illustration that is showing you your different stages of your cell cycle. And what we can do is we can either take a look at the illustrated example of the cell, which is the top part. We can also take a look at the animal histology. So this is most likely a whitefish blastula, just a little hollow ball of cell that we're looking at. And then obviously we can also take a look at the plant histology. And we know it's a plant because we see the really nice cell wall that is framing the cell, something that we lack. Now, in interphase, your main goal is to just go ahead and get through G1, S, and G2. And remind yourself that S is the part where you're going to go ahead and you're going to replicate your DNA. So we're now looking at cells that instead of having 46 chromosomes, have twice as many, they have 92. And the cell has twice as much DNA because it's anticipating going through mitosis and physically separating that one mother cell into two separate daughter cells. Each will have 46 chromosomes because we're trying to keep that number nice and consistent. The first phase of mitosis is going to be prophase. And in prophase, our main goal is to get rid of the nucleus. So we're going to see that the nucleus is going to start to disappear. Here's my little nucleus. The nuclear membrane is going to start disappearing. And we're doing this so that we can have more access to the chromosomes, which are starting to condense and they're becoming more visible. Now, a lot of books have started to introduce prometaphase. And prometaphase is sort of like a little phase between prophase and metaphase. This is usually where the end part of the nucleus breaks down, where the nucleolus also starts to break down. I'm going to be very honest with you. When we do our exam, we're just going to discuss prophase and then metaphase. I am not a big fan of prometaphase, but you might come across it when you do your homework assignment. So I definitely want to kind of highlight the fact that it talks about the tail end activities of prophase. For exam purposes, I just want you to know that prophase is going to be the part of mitosis where the chromosome become more visible. The nuclear membrane of the nucleus starts to break down, and those are really your two main events. Then you will go into metaphase. And metaphase is known for the fact that the chromosomes are going to align down the equator of the cell. So they're going to line up right down the middle of the cell. So in our case, all 92 copies that we have, because remember we doubled our 46, they're going to align right down the middle of the cell. And the reason they're doing that is because they want to go ahead and we want to split that number in half. We want to go back to our original cell count. So what we see happening is in anaphase, which follows metaphase, The centromeres that were holding together the sister chromatids, they will go ahead and break apart, and we see that the chromosomes start moving to opposite ends of the cell. Half of the chromosomes move towards the left, and the other half will move towards the right. So your sister chromatids will separate, and they'll create two separate batches at either ends of the cell. So for those of us keeping track, that means that 46 are moving this way and 46 are moving the opposite way. So we're back to our original number. And this is all happening in anaphase when the centromeres split and the chromosomes move over to opposite ends of the cell. And you can see it happening in our animal model, and we can see it happening in our plant model. To me, it always looked like fingers that were separating and going their individual way. The last part of mitosis is going to be telophase, and in telophase, we're going to have to do a few things. The first thing is that we need to reform our nucleus. So the nucleus reforms itself. So does the nucleolus. The chromosomes start to decondense, so they're not as tightly coiled up as they were before. We also see that our little protein spindle, which are these purple satellite features right here, they start to disappear. The protein spindle, which you saw in your previous pictures, they are basically our mechanical forces that we utilize to move the chromosomes from point A to point B. So in telophase, we see that the nucleus reappears. We see that the chromosomes start to decondense, so they're no longer as visible. And then at the end of telophase, we have an event called cytokinesis. Cytokinesis means the division of the cytoplasm. And the cytoplasm is the internal part of the cell, all this blue right here that you see. And in cytokinesis, what we do is we literally split the cytoplasm in half. And when we split it in half, we're able to generate two individual daughter cells. And each daughter cell will have its own nucleus. And inside the nucleus, you'll have 46 chromosomes, which is the same number of chromosomes that you had in the mother cell. So cytokinesis is division of the cytoplasm. It happens at the end of telophase, and it allows you to produce two daughter cells that are identical in genetic material from the mother cell that you had all the way in the beginning. Now, these two daughter cells will move right back into interphase, and they'll do it all over again. Now, I do want to point something out to you. When it comes to cytokinesis in plant cells and animal cells, we do see an indication that there's a slight difference, and part of that is because plant cells have to account for their cell wall division as well. So let's go ahead and take a look at the illustration that's on our next PowerPoint. And what we see happening here is that anytime that cytokinesis is occurring, you'll always have what we like to call a contractile ring, which is going to be the pinching off the cytoplasm that you start seeing right over here. And what we see happening is that the contractile ring will either go ahead and be referred to as the cleavage furrow or the cell plate. Both of these terms will indicate the location where the cytoplasm is going to split. However, you're going to use the term cleavage furrow if you're looking at an animal cell, and you're going to use the term cell plate if you're looking at a plant cell. And like I said, it's the same locational term, but one is just describing the fact that the animals simply have a plasma membrane, whereas the plants have a plasma membrane and a cell wall that they have to divide up amongst the daughter cells. So just to reiterate, if the cell is doing mitosis, your end product is that you produce two daughter cells that are the same genetic composition as the mother cell that originated everything on. Your body and the human body will utilize mitosis whenever we are reproducing our somatic cells. Our somatic cells are our non-reproductive cells, so they are what I like to call our everyday cells. So your skin cell, your hair cell, your stomach, your kidney, your large and your small intestines. Any cell in your body with the exception of your gametes, your reproductive cells, they are called somatic cells. And they will always have a diploid number. They will always have 46 chromosomes. And when those cells become damaged, or senescence, when they become old, they will need to utilize mitosis to form new daughter cells. Now, the other form of cell reproduction, your meiosis, you will utilize meiosis if you want to go ahead and you want to reduce the number of chromosomes you carry. So if you want to go from 46, which is diploid, or what we like to indicate as 2N, and I'm so sorry, my handwriting is horrible with this mouse. Diploid means that you have 2 copies, and that's because you get one from mom and one from dad, indicated by 2N, and that's why you could say you have 46 chromosomes, or you could say I have 23 pairs. So if you want to go ahead and reduce that number to 23, or haploid, meaning you only carry 1 copy, then if you want to do this process, you're going to have to do meiosis. Meiosis is going to allow us to reduce our genetic material, and it's going to allow us to produce 4 daughter cells, not 2. In order for you to do mitosis, you're going to utilize the same exact cell cycle, meaning that you're going to start with interphase. You're going to go through G1, S, and G2. In S, you will still synthesize your DNA copy. You'll go from 46 to 92. And when you now do your cell division, you will do your meiosis. And you're going to notice that we're going to utilize the same exact phases that we did in mitosis. So you're going to see terms like prophase, metaphase, anaphase, and telophase. But what we're going to do is we're going to do everything twice, because our end goal is to walk away with 4 cells that only carry half the genetic material of our original cell. So let's go ahead and take a look at prophase, metaphase, anaphase, and telophase in meiosis 1 and in meiosis 2. Alright, so first things first, remember everything will always start with interphase, because that is the day-to-day activity of the cell. And remember, within interphase, we're very interested in the S phase, because that is where our DNA is replicated when we go from 46 to 92 copies. I also want to remind you that in meiosis, our goal is to produce 4 daughter cells that will be haploid, so they'll have half the genetic material, and the only cells in our body that will utilize meiosis will be our gametes, our sex cells, so we're talking about sperm and egg cells. Now you're going to also notice that the phases are going to be in the same exact order, so you have your prophase, your prometaphase, your anaphase, your telophase, and the events are going to be the same. However, we're going to just repeat everything twice. So what I mean with that the events are going to be the same is in mitosis, I said to you, the main goal of prophase is to get rid of the nucleus and to condense your chromosomes and make them more visible. So notice right over here how it says meiosis, prophase 1. We're going to condense the chromosomes and make them more visible. And we're also going to start breaking down the nuclear envelope. So the same exact thing is happening. You're getting rid of the nucleus and your chromosomes are becoming more visible. But there's one thing that happens in meiosis that you rarely see in mitosis, and that is going to be this thing called crossing over. Crossing over happens in prophase of meiosis 1, and crossing over is when we swap genetic material between homologous chromosomes. So notice on the illustration right here, how they have indicated for you your blue and your pink pairing, your blue for the paternal, pink for maternal, and notice how the pink and the blue are swapping material. That is crossing over. And when you cross over, you allow the paternal and the maternal sections of homologous chromosomes to swap materials with each other. So you create almost like a little hybrid. Notice right over here on my illustration that my chromosome is mostly blue with a little bit of pink, mostly pink with a little bit of blue. And that is due to the genetic swapping of information that has occurred in crossing over. Now, crossing over, like I said, happens in meiosis. There have been cases that it occurs in mitosis, but is by far not as common as in meiosis. Now, metaphase will be, once again, that the chromosomes will align down the equator of the cell. Anaphase, we see that the homologous chromosomes will separate, and we see them moving to opposite ends of the pole. So for those of us keeping track of our numbers, that means 46 to the top, 46 to the bottom. And then in telophase and cytokinesis, you can see, just like we discussed in telophase and mitosis, we have the reformation of the nucleus. We see that the chromosomes are no longer as visible. And obviously, we see our little contractile ring start to form because we're splitting our cytoplasm. We're doing our cytokinesis. And right now, what we're going to do is we're going to divide the cell into two. Now, we're not done yet because meiosis tells us that we're going to do everything twice. So right now, we've done prophase 1, we've done metaphase 1, we've done anaphase 1, we've done telophase 1. And remember, in prophase 1, you had your crossing over where you swap genetic materials. So your sister chromatins are not 100% identical anymore. And at the end of meiosis 1, you have two daughter cells, and we're going to take those two daughter cells and we're going to repeat all the steps all over again. So on our next illustration, we see meiosis 2. And we can now also see that we have our daughter cells that we have produced. So these are the two daughter cells that we had at the end of meiosis 1. And we're going to see, we're going to do everything again. So we're going to do prophase 2. The DNA will once again become visible. The nuclear membrane will start to disappear. Metaphase 2, the chromosomes will align down the middle of the cell. Anaphase 2, the chromosomes will split. Now, the question is, how many chromosomes will you have on either end? Remember when we did our initial split, we had 46 in here, right? And then in metaphase, we line those 46 up. And in anaphase, we split the numbers. So that means that your 46 is now further reduced to 23. So you have 23 that's going to the left, 23 that's going to the right. And you're doing this into two daughter cells that you produced at the end of meiosis 1. Now, once you have your separation, you're going to go into telophase 2, which means I'm going to reform my nucleus. Once again, the chromosomes are going to become less visible. And once again, I'm going to do my cytokinesis, the splitting of the cytoplasm. And now what I'm going to see is I'm going to end up with 1, 2, 3, and 4 daughter cells. Each daughter cells will have how many chromosomes? 23, which is only half of what we started with. So that's our haploid copy. So notice right here how it says at the end of meiosis, you're going to walk away with 4 haploid daughter cells, and they're non-identical. The reason they're non-identical is because of the crossing over that has occurred. In fact, if you look very closely at your picture, you're going to notice that some of the chromosomes are fully blue and pink, and some of them have the recombination of crossing over that has occurred beyond them. So if you're going to do meiosis, you're only going to do it for your gamete cells, and it will allow you to produce 4 daughter cells that only have half the genetic material that you started with. And the daughter cells will be no longer identical from each other, and they'll also definitely not be identical from the mother cell that generated them. All right, so here's just another illustration showing us our crossing over. So we see our homologous pairs of chromosomes. Here's dad's version in blue, here's mom's version in pink, and we see in prophase 1, we have a chiasmata that forms, where it's that the sister chromatids, or I should say the non-sister chromatids from the homologous chromosomes will cross over, allowing you to go ahead and create a chromosome that is a recombination of both. So what we see happening is because part of the chromosome will be a new part or a hybrid, by the end of doing our meiosis, we are able to say that we either have what we call a parental or a recombinant cell. A parental cell means that the chromosome is fully intact as it originally was. So you can see right here fully blue or fully pink, because that's the original version that was inherited from the parent. The recombinant means that you've had crossing over occur and that it's now a combination of blue and pink. It is a combination of both parents. They did some statistics on this, and they figured out that with our 46 chromosomes, there are over 4 million different combinations that can be created, which means that you can go ahead and generate millions of different eggs and millions of different sperm cells. Some of them will be partially parental, and some of them will be partially recombinant. And this often can describe the fact that if you're looking at a family with multiple offsprings, how they look very similar, but they tend not to look identical. It all depends on which version of the gametes were utilized during fertilization. Alrighty, so let's go ahead and take a look at chapter 3. And, as we're perusing through our PowerPoints, if there's something that you're not really sure about, stop it, jot it down, go ahead and shoot me an email or go ahead and post it on the discussion session. But like I said, if there's something that I'm discussing and you want to read more about it, I would definitely recommend that you go ahead and you get the book. Sometimes I might assume that you have, that you're more comfortable with certain materials than you really are. And, we can all use a refresher from time to time. So don't be shy and utilize all the different resources that you have access to. Now, chapter 3 is going to go ahead and take a quick look at Mendelian inheritance. And I'm sure you guys have heard about Mendel before. There's this handsome picture that we see right there. We like to call him the father of genetics. He did a lot of work with pea plants, where he wrote about that you can predict the way the offspring will look and behave based on what the parental spring was all about. And he did all this without even knowing about the existence of DNA. So it is because of Mendel's work in the 1800s that we now have terms such as genotype and phenotype, diploid versus haploid, allele, and then of course dominant, recessive, homozygous, and heterozygous. Now, I've mentioned some of these terms before, so I just want to quickly make sure we're all on the same page. Your genotype is your collection of DNA, so these are your 46 chromosomes, and your phenotype will be your physical attributes or your behavioral attributes. So, for instance, if you were to give a description of yourself and you were to say, I am 5'8", I have green eyes, I have brown hair, I have fair complexion, what you're telling people is your phenotype. And the reason you have that particular color hair or eye color or skin tone is because of the genotype or the genes or the chromosomes that you've inherited. We have discussed the fact that we are diploid, meaning we get 2 copies of everything. Thanks, Mom and Dad. And then, of course, when it comes to our gametes, our sex cells, those are going to be haploid because you are only able to contribute half of your genetic information to your offspring. So you'll give 23, your partner will give 23, and your baby will have that magical number of 46 that works best for humans. An allele is variations of the same gene, and some alleles can be dominant, meaning that they'll always show through in the phenotype, while others are recessive. A recessive gene can be masked and tends to only show through if both parents contribute that, so if it is going to be a homozygous donation. Now, I'm also hoping that you guys are familiar with doing Punnett Squares. And if it's been a while since you've done a Punnett Square, I want you to go ahead and when you have some time, take a look on D2L in the Unit 1 folder. I'm going to post a little activity on setting up different Punnett Squares. So you can practice, separating the gametes and combining the maternal and the paternal to see what the potential offsprings look like. You do not have to be an expert in doing Punnett Squares, but you should have a nice little understanding of it because it will help you with some of the issues as we kind of look at some of the key terms for Unit 1. We're going to discuss monohybrids and dihybrid crosses as we move along, and then along the way we're also going to take a look at what we call the Law of Segregation and the Law of Independent Assortment, and of course the P, the F1, and the F2 generations. So let me just kind of show you this illustration right over here. I'm going to do a little circle around it. This is my good old-fashioned Punnett Square, and in a Punnett Square what you're doing is you're taking a look at what mom and dad are contributing in the form of their gametes. Now, since we are diploid organisms, that means we have 2 copies of everything. And the law of segregation says that when it's time to reproduce, we can only contribute 1 or a haploid copy of it. So over here you can see in my little Punnett square, I had a male gamete, and the T is going to stand for tall, because we're going to go ahead and steal Mendel's example of taking a look at pea plants. If the plant was tall or short, and tall or short, that's going to be the phenotype, the phenotypical description. The reason that the plant is tall or short will depend on which gene it inherits. So as you can see, what he's done is he's telling you that you have a capital T, which is going to be your tall. Oh my goodness, you guys, forgive me for this writing, it's horrendous. And then our lowercase t is going to indicate our dwarf or our short plant, okay? So if we are looking at a phenotype of being tall, that means from a genotypical aspect, your individual could either have 2 copies of the dominant, meaning a homozygous dominant, or it can have 1 copy of the dominant, 1 copy of the recessive. So it can be a heterozygous mix, which is the case right here in my little Punnett square. The male, in my example, has 1 capital T and 1 lowercase t. So that's what the male looks like. Now, the female is going to be the same. 1 capital T, 1 lowercase t. So when you look at the female, it's going to have a tall appearance. Now, the law of segregation says when it's time for division or reproduction to occur, we have to separate the diploid genes, and we have to go ahead and separate them into individual haploid options. So that means that for the male, it means that they either carry a capital T or a lowercase t. So I just separated these 2. And I do the same thing for mom. Capital T, lowercase t. So when I do my Punnett squares, and I know some of you know how to do this, so bear with me. When you do your Punnett square, you basically put the gametes of the male on one end and the gametes of the female on the other end. So one will go on top, the other one will go on the side. Doesn't really matter which one goes where, as long as you don't mix and match them. Now, the inside boxes are going to represent the kids. So if mom's capital T comes in contact with dad's capital T, child number 1 will be homozygous dominant. Homozygous means both are the same. Dominant means both of them are capital. So child number 1 will be tall. Child number 2 will be the capital T from mom with the lowercase t of dad. This will be a heterozygous mix. So what we would see from a phenotypical aspect is that the child will be tall. I hope everybody said that, because tall is the dominant one. Notice also how you always have to put the capital letter first before the lowercase one. Now, if we go to the bottom of our Punnett square, we have 2 more options. So we can see that now we have child number 3, which is also heterozygous, a capital T from dad, a lowercase t from mom. So also tall, having 1 of each. And then the 4th child is going to inherit 2 of the recessive copies. So it's going to be homozygous recessive. And this child will be short or have the dwarfism gene when it comes to our little pea plants that we are looking at. So if you're doing your Punnett square, you're basically trying to predict both the genotype and the phenotype of the children. And basically what this tells you is that if you have 2 parents that are heterozygous, you have a 25% chance of creating a homozygous recessive trait or a short child. And that is because it only means 1 out of the 4 boxes. And you have a 3 out of 4 or 75% chance that the phenotype of the offspring will be tall. So that's what little Punnett squares are. And I'll put a little practice activity for you in your folder on D2L so you can kind of play around with it a little bit more, and obviously just reach out if you need some additional assistance. To continue on right over here with the illustration that's all the way on the right-hand side, we see that the parental generation, this is the first original generation. You can go ahead and you can see the law of segregation has occurred where they separated. Now in this case what we're doing is we're mixing a homozygous dominant with a homozygous recessive. So as the alleles are separated, whenever you do your Punnett square, there's no other option but a capital T from one end with a lowercase t. So that means that the offspring in F1, our first generation, is all going to be heterozygous. They're all going to be tall, and they're all going to have one capital T and one lowercase t. And now what we're going to do is to create our second F2 generation is we're going to go ahead and we're going to do cross-fertilization. Cross-fertilization basically means that you're going to go ahead and do two of your heterozygous mixes. So capital T, lowercase t, times capital T, lowercase t, which is what we did right over here. And when you did that, then you're going to end up seeing that you have your gametes, just like we did last time. Capital T, lowercase t, capital T, lowercase t. And our four genotype combinations, this is very similar to our Punnett square. And right over here you could see your phenotypes. 3 of them will be tall, and 1 of them will be short or the dwarf. Okay? So this is just a look at what we call a monohybrid cross. Monohybrid means you're just looking at one trait. So in this case, we're just looking if it's going to be, what height is going to be, tall or short. You can obviously go ahead and build upon this. So for instance, if you're doing a dihybrid cross, that means that we're looking at 2 particular traits. So for instance, in this example, what you're doing is you're looking at the peas that are being produced, and you're looking at 2 different things about the peas. You're taking a look at the color, is it yellow or green? And you're also taking a look at the texture, is it round or is it wrinkled? So if you come over here, you can see that from a genotypical aspect, they're using the capital R's to indicate that round is dominant over wrinkled, and the capital Y's to indicate that yellow is dominant over green. Now, this one is a dihybrid, meaning we're looking at 2 different traits, and it also brings up what we like to call the Law of Independent Assortment. The Law of Independent Assortment says that if you're looking at 2 separate genes that are not located, that they are not linked on the same chromosome, that means that those 2 separate genes will go ahead and travel separate from each other. So what you need to do is you need to do some good old-fashioned foiling and think of all the different combinations that you can create between the R and the Y. In fact, if we come over here on the side, what we see happening is that if we have a cross where you have nothing but homozygous dominant and homozygous recessive that are being crossed together, no matter how you twist or turn it, all the gametes for this example with yellow will be round, so that's the capital R, and they'll be yellow, capital Y. So this is what all gametes are going to look like in our homozygous mix. In our homozygous recessive, what we see happening is that they will produce gametes that are all lowercase r for wrinkled and lowercase y for green. All right, so if you do your little Punnett square, your little Dihybrid square for that, we go ahead and we see that all offspring will inherit a heterozygous mix. Capital R, lowercase r, capital Y, lowercase y. And what we're going to do is we're going to do some good old-fashioned self-fertilization. So we're going to go ahead and cross a capital R, lowercase r, capital Y, lowercase y, with a capital R, lowercase r, capital Y, lowercase y. And when you do this, look what's happening over here. When you go to look at the gametes that you can create, you have to keep in mind your law of independent assortment. And that basically means that you have to think of all the different ways that the R's and the Y's can be paired up. So we can go ahead and do this thing called foiling, which basically is a math technique where you kind of keep track of your combinations. So what I mean with that is I can say that this capital R can go ahead and be paired with the capital Y. So here's my first mix. But the capital R can also be mixed with my lowercase y. There's my second option. And then I go back to my lowercase r, and I mix it up with my capital Y. That's my third option. And then my lowercase r can also be mixed with my lowercase y. There's my fourth option. So what I'm doing here is law of independent assortment because I am separating them. They are not on the same linked chromosome. The R and the Y move independently from each other. And notice as I'm doing this, I am also separating the gametes, meaning that I'm no longer carrying two of each of the letters. Only one letter of each R and Y is represented. You also want to keep in mind that when you're doing this, that it's often in alphabetical order. So that is why the R is posted before the Y. And also notice how when you have a dihybrid, you will have 4 gametes on each end, 4 different combinations, which means that when you look at your offspring, you'll have 16 little squares that you have to take a look at. And in fact, go ahead and take a look at the offspring. And what you're going to notice is as you're doing your offspring, you're basically just reuniting your 2 options for R and your 2 options for Y. Notice how the R's are paired and then the Y's are paired. So none of the offsprings would have, like, for instance, an RY combination. You don't do that because the genes are independent from each other. Instead, what you would do is you would say you have an offspring that is RR, YY. And that is because the genes are not linked. They tell you 2 separate entities. Is it round or wrinkled? Is it yellow or green? And an interesting thing to show you is that if you're doing a dihybrid square and both parents are heterozygous for both traits, you get a very stereotypical combination when you do your independent assortment. Let's go on to the next slide to show you what I mean with that. So right over here, we can see that we have our male and female gametes, and they are both coming from a heterozygous mix for 2 traits. And what we see happening is after we have added up all our 16 combinations, we see that we get our very stereotypical 9 to 3 to 3 to 1 ratio. This ratio is only seen when both parents are heterozygous. This is only seen when we're doing a dihybrid cross and also when we're doing an independent assortment. So the 2 letters are not linked to each other. The 9 indicates that you're looking at 9 round yellow seeds, or better yet said, you're going to have 9 that have the dominant for both traits when it comes to their phenotypes. The first 3 will indicate that you'll have 3 that will have a dominant and a recessive trait, and then you'll have another 3 that will indicate that you have a recessive and a dominant trait, and then the 1 indicates that you have your 1 option that will both carry recessive for both of the genes. So 9, everything is dominant. 3, 1 dominant, 1 recessive. 3, 1 recessive, 1 dominant. I know it's the same thing, just in a different order. And then 1 is that both of them are carrying the recessive traits. All right, so here you go. You can see it again on your little Punnett Square. This is going to be your dihybrid cross. Now, I will kind of give you a heads up on your exam. I will not ask you to complete a dihybrid cross because it will take up too much of your time. You will most likely come across a question on that on the homework, where you'll have more time and more opportunity to kind of explore it. On your exam, I will definitely be asking you, though, if I give you the genotype of the parent of the dihybrid, what the possible gametes are that they can produce. And keep in mind that when you're producing your gametes, you are doing your segregation, meaning that you're separating the 2 letters, and you need to have equal representation of them. So as I'm looking at my male and female gametes, notice how each of them will only have 1 representative of T, 1 representative of Y. So for instance, you'll never come across a gamete that will be like TT or YY. You need to have a representation of each of the 2 particular genotypes you're looking at. We can also take our crossings and we can put it in what we like to call a little pedigree. A pedigree is basically what I like to describe as a family tree. It allows us to see the offspring from one generation to the next. And we're going to be looking at quite a few pedigrees as we go along. So let me just go ahead and introduce you to some of the symbols that you're going to see. And don't get too overwhelmed. We're going to work our way up. But basically what you see happening is that the top layer will be the first generation. The square represents the genetic male, the circle, the genetic female. And then what we see happening is it will then tell you how many children they produce. So see how it's a little family tree? So that will be the children. And then it will start telling you when the children start mating. And it will then give rise to the grandkids. So here are the grandkids, and this will then be the third generation that's introduced. So this is mom and dad, these are the kids, and then these are the grandkids. And what it's showing you over here is, notice how some of the colorations are off. Let's say we are doing a study where we're taking a look at a gene that is sex-linked, or maybe a gene that's recessive, and the person will have the disease if they're dominant or heterozygous, whatever mix we're looking at. You notice that we can go ahead and we can utilize shading for that process. So for instance, if you see that the boxes are clear, usually we're looking at an uninfected individual. So from a genetic aspect, they're good to go. There's no issues. If you see that the boxes are fully shaded, that usually indicates that the disease is prevalent in that individual. So for instance, right over here, this offspring inherited the full disease. If you see that the boxes are half shaded, that usually indicates that they are looking at a disease that is heterozygous. And also what we see happening is that if the offspring only inherits one recessive trait or one disease trait, then they are then what we call a carrier. So they're able to pass it on to the next generation, but they do not have the disease. And we'll talk more about the pedigree, obviously, as we go along, definitely in a little bit when we take a look at sex-linked diseases. But for now, I just kind of wanted to show you it. And obviously, often there'll be the key right next to it, so you can kind of see which one is mom, which one's dad, which was the first generation, which one is the second generation. Alrighty, I just quickly want to mention statistics. I don't want to spend too much time on it because obviously many of you will go ahead and take a statistics class along the way. But obviously anytime you do research, there is some statistical analysis that have to come in play to see if there's any type of errors or mutations or any abnormalities that might be picked up on your data. There are many different ways to do statistical analysis. Probably the easiest is to do probability. The probability is basically when you take a look at the chances or the likelihood that an event will occur divided by the total amount of times that the event happens. So a good example would be, for instance, throwing a pair of dice and saying, you know, what is my probability of getting a 2 when I roll the dice? Well, you have 6 numbers on a dice. So if you're only looking to throw a 2, that means your probability is 1 out of 6. You have a 1 out of 6 chance that you'll do it. Or even more simple, if you do a coin flip, the coin has a head and a tail, so your probability that it lands on head is 1 out of 2. So just what's the probability of an event occurring out of the total amount of times that you're looking at it? You can also do more of a statistical analysis, which is basically when you take a look at more of a deeper analysis of it. So we have our probability. That one's very easy and straightforward. But then you also have what we like to call a binomial expansion equation. This is when you start to look at all the possibilities in an unordered event. So for instance, if I say to you, what are the chances that if I have 5 kids, 2 of them will be redheads? Or what are the chances that if I have 2 dice, I'm going to roll the 3? What are the chances that if I flip 5 coins 2 times, I'm going to have heads? So that's what a binomial extension takes a look at. It takes a look at what are your possibilities out of the cumulative, but in an unordered event. So you just want to know what are my chances of X out of so many times when I'm doing it. Most of the time, if you're into serious data set collection, you do what we call a chi-square test. And this is your goodness of fit. It's a statistical method where you're basically comparing your hypothesis, what you expected, to what your observed values were, and you see how good they fit within that particular hypothetical explanation. When you do your chi-square value, oftentimes you have to calculate your degrees of freedom. Your degrees of freedom will often take a look at how many of the different trademarks you are looking at. And once you put in your number for your variation between expected and observed, you would go ahead and you would generate a p-value. And the p-value would tell you if you were to accept or reject your hypothesis. Usually the wider the gap is between observed and expected, the more likely it is that you're going to be rejecting your hypothesis. And that usually indicates that there's some abnormality trend, which might be a good thing or a bad thing, because you might be looking for something that's a mutation, or it can also indicate for you the fact that there might have been some experimental errors and that the data set needs to be repeated. Go ahead and reference the book if you want to read a little bit more about statistics, but I just wanted to kind of give you a nice little, really like a few seconds intro of probability, your binomial, and your chi-square test. And obviously if you want to chat more about it, that's perfectly fine with me. Let's meet up in the discussion sessions. I am actually going to end this particular part of our first unit. I see we're a little bit over the 1 hour mark, so go ahead and make your notations, and then I will see you in a little bit whenever you're ready for the second part of unit 1, excuse me.